The overall goal of the following experiment is to detect antibodies against adventitious infectious agents in laboratory animals. This is achieved by incubating test serum with antigen-coated microspheres. Next, the serum is incubated with a biotinylated tagged, species-specific anti-immunoglobulin, which detects any antigen-antibody immune complexes. Then, a streptoavidin-labeled phycoerythrin fluorophore solution is added in order to detect any biotinylated immunoglobulin. Results are obtained that show positive or negative antibody status for common agents in lab rodents based on multiplexed fluorescence immunoassay net score fluorescence sample values.
The main advantage of using this technique over the existing method like ELISA is that MFIA is a multiplex assay. This enables a researcher to screen up to 100 different assays in a single test well. This technique decreases the amount of serum, reagents, and disposables used in the existing methods, while increasing the amount of information generated from a single test well. We first had the idea of using this method when we were testing a large number of sera samples for multiple agents and wanted to reduce labor as well as testing time. Demonstrating the procedure will be Donna Cohen, a senior technologist from our lab.
To begin, have ready 2 buffers required for the multiplexed fluorescence immunoassay, or MFIA procedure. The first is a primary diluent, which is available from Charles River and contains proprietary blocking agents to decrease nonspecific interactions. For the assay wash buffer, prepare a solution of PBS with 1% BSA.